Hello grade 8 students, welcome to a new lecture and we are still talking about the non-specific immune response. In the previous lecture, we learned that the immune system is responsible for defeating our bodies against non-self. And we learned also that the natural barriers are the first line of defense against any foreign pathogen. Natural barriers such as tears, nasal secretions, skin, saliva, gastric juice, mucus and ciliated cells, and many others. But if these natural barriers fail to prevent microbes from entering, we have the second line of defense, which is the inflammatory reaction. The inflammatory reaction has some signs like redness, heat, pain, and edema. If these signs are present, then there is inflammation. And a part of this inflammatory reaction is a process known as phagocytosis. Today we're going to talk in details about phagocytosis. What is phagocytosis? Phagocytosis is a non-specific immune response ensured by granulocytes and macrophages. During this process, these cells come near to the microbe, adhere to them, engulf and digest them without identification of their nature, because they can't identify their nature, so it is a non-specific immune response. Okay, so during inflammation, phagocytosis took place where some leukocytes, phagocyte means eat the bacterial cell, directly without recognizing or binding to its antigen. Because of that, the response is not mediated against a specific antigen, but against all foreign bacteria entered. Therefore, it is called non-specific immune response. This process consists of 5 stages: attraction, then adhesion, then engulfment, then breakdown, and then release out. Okay? The next document represents these 5 steps, but are organized randomly. So these are the stages of phagocytosis, but they are arranged randomly here. Part A, name each of the above stages. Okay, to make it easy, let's look for them one by one. So where is attraction here? Attraction is in B. Why stage B is attraction? Because the phagocyte is attracted to the foreign body, to the pathogen. Okay? Let's look for adhesion. Where is the phagocyte getting very near from the pathogen? It's E. Then engulfment. Where is the phagocyte eating the foreign body? It's C. Engulfment is C. It's eating or engulfing the pathogen. And then we have the breaking down. Where is the phagocyte breaking down the pathogen? It's D. Look at it. It's breaking it down and finally releasing out. After breaking it down, the phagocyte kills it out and releases it out. So it's A, release out. Rearrange the above stages. So we know the stages in order are attraction, adhesion, engulfment, breakdown, then release out. So first one is B, then E, then C, then D, then A. Indicate that the shape of the nucleus of this leukocyte. Look at the phagocyte or the leukocyte here. How is the shape of the nucleus? As you see, it's horseshoe-shaped, the nucleus. Conclude the name of the given leukocyte. What is the name of the leukocyte that has a horseshoe-shaped nucleus? It's for sure the macrophage. Name another leukocyte involved in this process. Okay, so the two leukocytes involved in phagocytosis are macrophages and the granulocytes. So the other one is granulocytes. Part F. This response is classified as a non-specific response. Justify the statement using the given text. Okay. So, the response is not mediated against a specific antigen, but against all foreign bacteria entered. So it doesn't depend on the identity of the intruder. That's why it is a non-specific immune response. Let's solve application 1 together. Our bodies are equipped by different strategies of defense against viruses and bacteria. In the field of studying the role of skin in this issue, we realized the following experiments. So we have 3 mice and 3 different experiments. Let's see part A. Indicate the objective of these experiments as written in the short text here. The objective is to study the role of skin in defense against viruses and bacteria. Analyze experiments done on mice A and B. Okay, so what did they do? Look, in mouse A, on the intact skin, they place a layer of bacteria on this skin. Intact skin is the healthy or the not injured skin. The result was it remained healthy. Whereas in mouse B, the skin is injured and they place a layer of bacteria on its skin. It became sick. So let's analyze it together. Analyze, we can start from the variable then the result, or from the result then the variable. Let's start from the variable. So we will say that when a layer of bacteria was placed on the intact skin of mouse A, it remained healthy. Whereas, to connect them, whereas when this layer is placed on the injured skin of mouse B, it became sick. Conclude concerning the role of skin. The skin, as we know, it is a natural barrier. So we say, I conclude that the skin acts as a natural barrier. Using your acquired knowledge, explain the observation in mouse C. What did they do in mouse C? Mouse C, they introduced bacteria with the food to the mouse, to its intact skin, and it remained healthy. So the bacteria found in food was destroyed by the gastric juice of the stomach. That's why it remained healthy. The next figure shows the injured skin of mouse of experiment B. Note, chemotactic factor is a factor that attracts the white blood cells to the infected tissue or to the bacteria. Okay, part E, identify the leukocyte involved in this figure. You can see the leukocyte here, it has a multi-lobed nucleus, so it is a granulocyte. So, identify, we have to start from justification. So we say, since it has a multi-lobed nucleus, so it is a granulocyte. Name stage A. Look at stage A. What is stage A here? It crossed the capillary wall, and look at stage B, 2. It is phagocytosis, as you see. Pick out the factor that attracts leukocytes toward the bacteria. The chemotactic factor that is mentioned in the above text. Conclude the name of this reaction. What is the name of this reaction? We learned before, it is the inflammation reaction or inflammatory reaction. Identify if this response is a specific one or not. We say, since granulocytes engulf bacteria without recognizing and binding to its antigen, then it's non-specific immune response. Detailed study showed that leukocyte has to add some enzymes over the bacteria in order to digest it. The figure beside shows 3 stages A, B, and C of stage number 2 in the random order. Okay, so stage number 2 was phagocytosis. So A, B, and C are some stages in phagocytosis process. What is A? A, as you see here, is the granulocytes. The granulocyte gets very near from the pathogen, so it's adhesion. B, the phagocyte here, granulocyte, is eating or engulfing the pathogen, the bacteria, so it's engulfment. C is breakdown, because the granulocyte is breaking down the bacteria. In order, they are adhesion, then engulfment, then breaking down. So A, then B, then C. During this process, the following events took place. Fill the tables in the suitable clinical sign obtained. High concentration of cells push on the nerve endings. It's pain. It causes pain in the inflammatory reaction. These are the signs of the inflammatory reactions. Blood flows rapidly to the injured area. It's redness. Blood flow makes a friction with the capillary wall. It's heat. A lot of leukocytes cross the capillary wall toward the infected tissue. It's edema. In some rare times, process 2 fails. Formulate 2 hypotheses concerning the origin of this failure. Hypothesis 1, the enzymes necessary for digestion are absent. Hypothesis 2, bacteria become resistant to the enzymes. Predict in this case the cells that will interfere to save the body. Lymphocytes B and T may interfere to save the body. So that's all about today's lecture. Thank you for listening. Bye bye.